Started, so it's my great pleasure to welcome and introduce our seminar speaker, Kirk Lohmueller. Kirk got his undergraduate degree from Georgetown University, and then he did his PhD in Cornell, working with Andy Clark and Carlos Bustamante. After that, he did postdoc in UC Berkeley with Rasmus Nielsen as a Miller Fellow. So then he started his independent lab at UCLA, where he is now a full professor. He has also been the director of their bioinformatics program and the vice chair of their EEB department. So Kirk's research career started when he was a high school student, and his best cited paper was based on his high school research. Can you believe that? So during his high school, he did research in Whitehead Institute in MIT, and he was working on a meta-analysis of association studies, GWAS. Okay, so that was the very early days of GWAS, and so there were a lot of uncertainties, a lot of doubts about GWAS. So some people reported associations, others couldn't replicate. Okay, so does that mean GWAS was wrong, or does that mean there were sample size issues, etc.? So Kirk actually did a meta-analysis and demonstrating that there were true signals in GWAS. So GWAS was promising, and that turned out to be true. And that paper was cited more than 2,000 times. So Kirk has always been interested in population genetics and in deleterious mutations. So when he was a graduate student, he famously published this paper in Nature where he showed that there are proportionally more deleterious mutations in European population than in African populations. In his own lab, he collaborated with Robert Wayne on dog domestication and the population genetics of dogs and wolves, and they studied the consequence of small populations in terms of the inbreeding and deleterious mutation accumulation, etc. So today he's going to talk to us about fitness effects of mutations and implications for small populations. Let's welcome Kirk.
All right. Thank you, George, for the introduction, and thank you all for being here. I've had a great visit, and you have a really wonderful community here. So I'm going to start with a cliche, and that is that mutation is the ultimate source of genetic variation. And while it's a cliche, it turns out it's actually true. All of the genetic variants that we may see segregating in a population or that natural selection may be acting on, at one point in time came about as a result of the mutational process. And it's been long appreciated that mutations can have different effects on fitness. Some mutations, like the transposable element insertion in the classic peppered moth, may be beneficial, conferring a fitness advantage in those individuals that carry them. Other mutations may have a phenotypic effect, but may not actually affect fitness. And those mutations may be neutral with respect to evolutionary processes. Still yet, other mutations may be deleterious, and those would be mutations that result in either a decreased probability of survival and or decreased probability of reproducing in subsequent generations. So one of the key questions in evolutionary genetics is understanding the relative proportions of mutations in the genome that fall into these different categories. And so population geneticists in particular have formalized this idea in something called the distribution of fitness effects, or DFE. And this is a central quantity to what I'll be talking about today. And so what the DFE is simply a probability distribution, or you can think of it as a histogram, essentially reflecting if you put mutations randomly into either the genome or some compartment of the genome, what would their selection coefficients be? And so here's one hypothetical distribution of fitness effects that has a reasonable proportion of neutral mutations, some strongly deleterious mutations, and some in the middle. Another hypothetical distribution of fitness effects could look something like this, where there's a lot of weakly deleterious mutations, and yet another DFE could include a proportion of beneficial mutations as well. So there's tremendous interest in trying to infer what this distribution looks like for different types of mutations in the genome. And so the approach that my lab has taken to tackle this question is to essentially use genetic variation segregating in natural populations combined with population genetic models of the evolutionary process to try to back infer what the DFE looks like. So just to say a few more words about that, the genetic variation we see in sequence data from individuals sampled from the population, it's essentially the combined, represents the combination of what this initial DFE of new mutations looked like, as well as what the effects of selection and drift did to those mutations before we sampled them in the data. And so what we're going to do is start with this sequence data, use models of the population genetic and evolutionary process to then try to back infer what might this DFE look like. So it's essentially running this process back in reverse. And so in my talk today, I'm going to tell you about 3 vignettes related to fitness effects of mutations. And so the first is, what does the distribution of fitness effects look like for amino acid changing mutations, and how does it vary across species? Second, I'll talk about some of our new and ongoing work on the DFE for non-coding mutations in the human genome. And then lastly, I will sort of shift a bit away from talking about inferring the DFE, but thinking about, okay, what do deleterious mutations do in small populations, and how can we understand how they affect the fate of these populations? So that's the plan for today. And first, I'll get started talking about how we estimate the DFE for new mutations and how it varies across species. So a key concept here that we need to be familiar with for the next few parts, or for the first few parts of my talk, is the summary statistic of genetic variation called the site frequency spectrum. So imagine we sequence some DNA. This could be, for example, a coding region of a particular gene, and the red boxes indicate the genetic variants that we see there. So what we could do is simply tabulate the allele frequencies of each of these mutations. So here, at this first site, the derived or mutant alleles at frequency 1 out of 5. At the second, it's 2 out of 5, and so on and so forth. And so the site frequency spectrum is just a histogram summarizing these frequencies. So for example, there are 5 mutations in this data set, or this region of the genome, where the mutant allele is at frequency 1 out of 5. There are 2 where it's at frequency 2 out of 5, 2 where it's at frequency 3 out of 5, and none where it's at frequency 4 out of 5. So what we can do is simply tabulate this statistic from our empirical sequencing data, and that's a nifty way to summarize it. But we can also, and I'll talk about this in a second, calculate what this site frequency spectrum would be from a given population genetic model. So for a given set of mutational parameters, demographic history parameters, and selection parameters, we can predict what do we expect the frequency spectrum to look like. And so here I'm showing you site frequency spectra from a population genetic model, where here all mutations are neutral, and over here on the right side of this plot, they're all strongly deleterious. And so you note that the frequency spectrum is quite sensitive to the effects of selection. Whereas mutations become more deleterious, you notice that there's just fewer of them segregating in the sample. And of the ones that are still there, they're almost all at low frequency. And intuitively, this is what you would expect to see, right? Selection is removing deleterious mutations from the population, and those that aren't quite entirely removed, they're just kicking around at really low frequency. And so this statistic is the site frequency spectrum is quite sensitive to the effects of negative natural selection or to the presence of deleterious mutations. Now, there's a little bit of complexity in how we actually do this, because the demographic history, like whether the population expanded or contracted, also can affect the site frequency spectrum. And so the approach that we're taking here and that others have taken is to use a class of mutations that are thought to be more putatively neutral. And I know George will probably give me a hard time about this at dinner later. But in this case, synonymous mutations, where those mutations where the changes to the DNA sequence don't change the encoded amino acids, we estimate demographic parameters from the frequency spectrum at those sites, and then conditional on those demographic parameters, we then ask, okay, what are the parameters of selection or of the DFE that can match then the non-synonymous frequency spectrum or the amino acid changing mutation frequency spectrum. So that's kind of the overview of the approach that we're taking. And I glossed over some key ideas relating to, okay, I mentioned we can have our parameter estimates, and we can determine what our parameter values, and we can determine what the site frequency spectra will look like. Well, to do that, we need a population genetic model. And the model that we're using is the Poisson random field model developed by Sawyer and Hartle. And basically, the idea behind this model is fairly simple, and that is that mutations are assumed to enter the population via a Poisson process, and then each mutation follows its own Wright-Fisher model of drift and selection. And so then the site frequency spectrum is just the sum of all of these together at the end. And there's some mathematical tricks that go into actually working with the Wright-Fisher model using diffusion approximations to really make all of this tractable, but that's the essential idea behind what we're doing. And so one of the an early graduate student in my lab developed a series of Python modules to actually implement this approach for inferring the DFE called FitDadi. Where DADI is diffusion approximation for demographic inference, and we didn't come up with that part, but the fit part refers to the fitness effects of the mutations. And so when Bernard applied this method to data from non-synonymous mutations in humans, this is what we had inferred, where these are different bins of strength of selection. So these would be the mutations that are close to being nearly neutral, and these would be the more strongly deleterious ones over here. So this is essentially discretizing that continuous distribution into something that's a little easier to see. But what you can see, in fact, is that about maybe 25% or so of mutations in humans are essentially nearly neutral. A bunch are in the weakly to moderately deleterious category, and then somewhere about 12 to 15% are thought to be, or we estimate to be, strongly deleterious. And this is consistent with other estimates of these proportions that have been made with other data sets and using slightly different approaches. But that's great. That's humans. That's one organism. I think a key question is, well, what if, do other species have essentially the same DFE, or do these proportions of mutations that are deleterious, how do they vary across species? And so my group's been working on this question for a number of years, and I'll tell you about some semi-published ongoing work that is done by Mei-Shi Lin, who is a former grad student with me, and Bob Wayne, currently a postdoc at UC Berkeley. And what Mei-Shi did was leverage a number of different data sets to estimate the distribution of fitness effects using the same approach I've been telling you about in these 11 different species, ranging from a number of insect species, 2 species of birds, and a variety of different mammals, including humans here as well. And so why these species? Good question. We wanted to focus on data sets that had a number of individuals sequenced from roughly the same population, however that can be defined, and that where the genomes were to a reasonably, I'd say moderately high to high coverage. So that was the key feature that we were looking for, to include different species. There's a lot of bioinformatic work and quality control that went on that I won't have time to tell you about here, but it's described in the bioRxiv preprint. But just to give you a sense of kind of what this looks like before I jump to the results, this is for one of the flycatchers and the wolves here. We have our synonymous, non-synonymous. This is plots of the frequency spectra, but now instead of the bars, it's as these lines. And where we have the empirical ones in blue, and then what we from our best fitting models here, just including demographic history and mutation, and then here including for the non-synonymous mutations, the demographic processes, but then also building in the model of negative selection acting on the deleterious mutations. So we did this for all of the 11 species, and here's what we found. So let me walk you through this plot. So the different species are shown here, somewhat phylogenetically organized. And on the x-axis is our best estimate of the average strength of selection. And the numbers on the points indicate the relative rankings of the species relative to one another. So with 1 being the lowest and 11 being the highest. And so you note a couple different things here. So first is you may note the wolves tend to be towards the high end, in fact, the 2 highest values. And that's actually why we included 2 different wolf data sets, because originally we had 1, and we're a bit surprised at how large the estimate was. And so we obtained another independent data set and saw similar results. But what you notice here is that the insect species tend to have lower average strength of selection acting on deleterious mutations compared to the mammals up here, and birds are somewhat intermediate to the insects and mammals. So that was just looking at summarizing the distribution with just the expected value. This is like a histogram that I showed previously from Bernard's paper, but now with a whole bunch of species on here. And this is indicating, again, the proportions of mutations estimated to be nearly neutral versus strongly deleterious down here. And the different colors represent the different species. And so what you really see is in the insects that they tend to have more higher probability of these mutations here with S of 10 to the minus 4th to 10 to the minus 3rd, and fewer down in the more very strongly deleterious category. So just to point out that what the results I showed you were assuming that the DFE is a gamma distribution, a gamma probability distribution, but that's largely a matter of convenience. So we explored other probability distributions, including a mixture distribution with a point mass on neutrality, log normal distribution, although this didn't actually fit the data as well. And you can see that there's some variation in the parameter estimates depending on the functional form assumed. And so what we did is, what I'll tell you about moving forward is actually using some model averaging, model averaged estimates where we essentially average these but weighting them by their relative fits to the data. Another thing we were really concerned about is does our method to estimate the distribution of fitness effects, does its performance vary with, say, population size, for example? And I'll talk more about population size in a minute. But one thing we were interested in is whether in the insects, inferring a much smaller average selection coefficient, could that be an artifact of just the population size being so large? And so what we did was carry out some slim simulations where it's kind of what I call in silico CRISPR, where we took the Drosophila demographic history, so the really big population sizes, but put in the human distribution of fitness effects in terms of S, and then simulated that and asked, okay, do we get estimates of the DFE that are more similar to the human one, or do they look like the Drosophila one, which is not what they should be? And so here I'm showing you the true value for humans, or when I say true, the assumed one in the simulation. This is what we assumed for what Drosophila would be for the DFE, and these are our estimates of the shape and scale parameters of the gamma distribution. So you can see they're clustering around the human value with some variance, and then here's, if we look at the average selection coefficient, you can see that, again, with some uncertainty, they are clustered around the human value, suggesting that seeing the small average selection coefficient in Drosophila isn't just due to a limitation of the method with large population sizes. So we also did a more formal model comparison across the different species to see, do the gamma distribution parameters seem to differ across pairs of species. So we could do that in a series of likelihood ratio tests, where in the null model assumes, like, say, Drosophila and fin whale, the two shown here, have the same parameters versus one where each species is allowed to have its own DFE. And so this is showing some likelihood surfaces for the constrained model shown by this star here, which is what we would infer if, say, fin whale and Drosophila have the same parameters versus a model where fin whale can have its own and Drosophila can have its own. And in fact, we infer that there's a pretty big difference or improvement in log likelihood when the species can have their own DFE. So we did this game of pairwise comparisons of comparing essentially pairs of species. And what I'm showing in this heat map is essentially the color indicates the strength of the difference. And so red means the species tend to have more similar DFEs, whereas white and yellow indicate that they're more different to each other. And importantly, this is made without reference to any phylogeny at this point. But what we see is, for example, that mouse and humans tend to be more similar to each other. Mouse and, say, Drosophila down here tend to be extremely different. Mosquitoes and bees seem to be more similar. Some of the mammals seem to be a bit more similar to each other. And in fact, if you basically ask, okay, take all these pairwise tests and ask, are they more different for species that are in a different class or the same class, we see that actually for species that seem to be more phylogenetically diverged seem to have more different DFEs, or the likelihood ratio test statistics tend to suggest they're more different. Of course, we could try to quantify this a little bit more formally, this phylogenetic signal, by actually looking at the covariation between the DFE parameters with the phylogeny. And what we find here is, using Pagel's lambda, we see that it's pretty large, of 0.84, suggesting, in fact, strong covariation of the DFE parameters with the phylogeny. So we next wanted to try to better understand, well, why. So the DFE seems to be changing over time and seems to be covarying with phylogeny, but why might that be the case? And so one thing we wanted to try to do is to see, well, are there different life history traits potentially associated with the different fitness effect, or with the different average strength of selection? And so one thing population geneticists always will look at for pretty much anything is long-term population size, or NE. And in fact, we see that there's a weak negative correlation here between the strength of selection and long-term population size, where in really large populations, the mutations tend to be less deleterious compared to smaller populations where they tend to be more deleterious. However, the effect is modest, and the strength of that effect is not super strong. We saw a stronger positive correlation with body mass, actually, where the more larger organisms tended to have more deleterious mutations. But I want to go back actually a second to the population size, that this effect, I mentioned it was weak, but beyond that, you notice these are the canids here that seem to greatly depart from the pattern. And if you look at sort of this spot here of a long-term population size of around 100,000, you can see there's a number of different species with population sizes in that ballpark with pretty striking variation in the average strength of selection, suggesting, in fact, that population size isn't the whole thing accounting for this difference. And I don't believe I have the results in here, but we actually looked for the population scaled selection coefficient to see, did that differ across species, and it also did. So it's not just that the N and S are canceling each other out. Whoops, sorry. So we looked at some more life history traits like generation time or mutation rate, and these we found actually weren't associated after correcting for phylogeny. So at the end of that, we don't really have a super strong association, I think, between any of the life history traits. Maybe body mass index, or excuse me, body size may show the biggest effect, but still it's modest. So in sum, for this part of the talk, the DFE seems to vary across animals, with mutations being more deleterious in mammals than in insects. These estimates of the DFE tend to be fairly robust to different things that we've looked at, and we looked at more than what I had time to essentially tell you about. But we see strong phylogenetic signal in the DFE variation at this deep phylogenetic scale. Some evidence of long-term population size being negatively correlated, though it can't explain all of the variation. And I think what this points to is the need for further comparative genomic work, perhaps sampling more deeply within specific clades that may vary in different traits or different population sizes and other attributes to really try to get at the factors disentangling or disentangle the factors driving the difference in the DFE across species. So far, what I've told you about has been very much focused only on non-synonymous mutations. And it's well appreciated that in mammals and in humans, right, that most of the DNA doesn't code for protein. And you know, the ENCODE project and other large-scale projects have been trying to annotate and determine the function of all this non-coding DNA. And so a natural question is, well, what are the fitness effects of mutations in the non-coding human genome? And I would say this has been a challenging question because the annotation of the non-coding genome has been much more challenging. And really, these large-scale projects like ENCODE have produced a lot of data to better annotate the genome, but it's still challenging. And so this is where my collaborator at UCLA, Jason Ernst, comes to the rescue, where he developed a computational method, Chrome-HMM, that can essentially integrate these multiple functional genomic data sets into an annotation that's a bit more biologically interpretable and helpful for actually annotating the genome for these sorts of evolutionary analyses. And I should point out, this work I'm about to tell you about is from Chen-Lu Di, a postdoc in my group. And so Chrome-HMM, the method that Jason developed, is you take these different epigenomic profiles along the sequence, and it's basically a hidden Markov model that goes along the genome and takes the multivariate profile of these different signals as what gets emitted from the different states. And then essentially what it will do is classify the genome into these different states that tend to be associated with different biological functions, so like active transcription start sites or transcription enhancers or repressed sorts of elements. So we could essentially annotate the genome using Chrome-HMM and then intersect these annotations with the genetic variation to try to understand what's happening. But one thing that question I get asked a lot is, how does mutation rate vary with these different chromatin states, right? Because there's different biology happening in different parts of the genome that's likely to influence how mutations happen. And so what we did here was took estimates of the mutation rate from Shamil Suniev's group using their roulette method for estimating mutation rate at a really fine scale across the genome. So that's what I'm showing here on the y-axis. And then these are different groups of chromatin states. Just to point out a few, this is quiescent. We've got DNase, hypersensitive sites. Let's see, heterochromatin here, transcription start sites. And so what you can see is, in fact, that there is variation of estimated mutation rate with the functional genomic profile. Is it a little? Is it a lot? I don't know. I mean, it seems slight, but it could be enough to make a difference. And so in the model estimation work I'll be telling you about in a few minutes, we actually include this estimate of the mutation rate for in our modeling, so that we can account for this variation or covariation of mutation rate with the chromatin state. So to give you a bit of a sense of kind of what the genome looks like, I'm showing you here a plot of just genetic diversity calculated. This is from the high coverage 1,000 genomes Yoruba. Each dot here represents a particular chrome HMM state. So there's like 100 of these different states. The colors represent these larger groupings of states together. That is admittedly somewhat arbitrary. So for example, the green ones refer to regions that seem to be transcribed. And by the way, I should point out, for everything I'm telling you about here, we've taken out the coding regions from the genome. So this is all non-coding stuff here. And so you can see is, right, these are the ones that have, the green states tend to have a bit lower genetic diversity. Red are the, like by the promoters and transcription start sites, so lower diversity, maybe some lower mutation rates. Heterochromatin is up here in yellow, quiescent in gray. They kind of cluster together, which is kind of interesting and seem to show higher genetic diversity. Enhancers are somewhat kind of in the middle. If we then normalize the genetic diversity by the mutation rate, that's what I'm showing here on the x-axis, and then plot on the y-axis a summary of the site frequency spectrum to G minus D, this is what you see. And the important thing to note is negative values of D indicate a skew towards lower frequency mutations. And so what I think is really striking here is that, so red is promoter and green are transcribed states. They tend to be the ones that have low genetic diversity and more negative to G minus D. And so these would both be signatures of negative selection, where there's a reduction in variation and skew towards rare variants. Whereas up here you see in yellow the heterochromatin and the quiescent states in gray tend to have less of a skew in the frequency spectrum and more diversity. So our interpretation would be these are the more neutral states, and stuff down here is the effects of negative selection. I should point out the enhancers are in blue are kind of in the middle of this. So they look less selected than promoters, but less neutral than the quiescent or heterochromatin. So we think from this, there's some evidence for selection contained within the data sets here. But this is, of course, very qualitative, and I'm just kind of pointing this looks down here compared to up there. So we wanted to look at this a bit more quantitatively through estimating the distribution of fitness effects of these new mutations using an approach like we did for the amino acid changing mutations. But the non-coding genome is a dangerous place, because I mentioned that we need some control regions to infer the demographic history. And so how do we do that here? Because there's a few things we're worried about. If this is the genome, right, you've got your coding regions in red, are, say, target non-coding regions. So these would be like the enhancer states, for example, in blue. And then we've got these quiescent regions in gray. And so this is what the genome looks like. And it's possible that the coding regions are influencing the target regions and the quiescent regions, through link selection, as well as we have the real effects of, or direct effects, I should say, of selection on the target non-coding regions. And so it's kind of a mess to disentangle. And so the approach that Chenlu took was to take, for each of our target non-coding regions in blue, is to basically take a window surrounding it and use the quiescent sites within that neighborhood to be the control sites for this particular chunk, if you will, and do that repeatedly across the genome. And so what Chenlu would do is take the frequency spectrum from these quiescent sites nearby, fit the demographic model, and would, take the annotated genomic region, the target region, find the quiescent control, fit the demographic model to this quiescent control, and then go back and take the frequency spectrum from the target region, use this demographic model, and then infer the DFE on top of that. And so when we do that, this is the picture that we see. So this is another one of those bar plot type plots where, again, these would be the more neutral mutations. This is very strongly deleterious, and the middle represents the intermediate strengths of selection. And so what I'm showing you here are for the Chrome HMM annotated enhancers in blue, promoters in red, and previous estimates of non-synonymous mutation, the DFE for non-synonymous mutations that I showed you previously. So you notice a couple things. First of all, the proportion of neutral mutations in the non-coding genome, or the functionally annotated non-coding genome, is much higher than that for non-synonymous mutations. That, I think, was entirely expected. And similarly, there's fewer of the more strongly deleterious mutations in the non-coding annotations compared to non-synonymous mutations. Again, I think expected. But if we now compare, say, enhancers and promoters, what you see is that promoters have fewer neutral mutations than do enhancers. And the promoters seem to have, or I should say, annotated promoters tend to have more moderately deleterious mutations than do the enhancers. And in the enhancers largely are mostly neutral, but there's a small proportion that are non-neutral. And it's harder to get an estimate of the precise proportion there. If you don't like Chrome HMM and you like ENCODE and their cis-regulatory elements, I got you covered here too. We repeated this analysis actually using the ENCODE definitions of promoter-like sequence and enhancers. And that's what's shown by the slightly lighter colors here. And they're pretty concordant in terms of the DFE estimates, despite the fact that the base pairs that are annotated actually are a bit less concordant than maybe one would have thought between the Chrome HMM and the ENCODE enhancers. So this at least gives us some sense that the estimates are a bit robust to the specific annotation that's being used here. So, okay, functional genomic data is one way to annotate a genome. Another way is through looking for regions of conservation across divergent species, right? And so the idea is if you take a lot of species, line them up at positions that are not targeted by selection, you might see a bunch of variation across species, but at bases that are important where negative selection removed variation, you might see the same, you might see an absence of a variation, right? And this gave rise to the whole comparative genomics paradigm that's been, I think, quite important at helping locate some of these regulatory regions, et cetera. And various methods using various different approaches, roughly say about 5% of the non-coding genome has been conserved across distantly related species, and that's implying negative selection there and biological function. So we wanted to essentially evaluate what are actually the proportions of deleterious mutations in these constrained regions, and in constrained regions that may be putatively functional. So maybe not getting down to enhancer versus promoter, but looks in the functional genomic data to have some sort of function. And so I'll show you some results now from using FastCons scores as our comparative genomics tool. And here the alignments are focusing exclusively on primates. We've looked at other alignments and annotations using all mammals, using different scores. Happy to talk more about that after because there's some interesting differences, but I'll give you the gist of it here. And so what we did is say, okay, if we take the top 5% of the non-coding functional genome from FastCons, let's infer the DFE for that. And that's what I'm showing now just in this sort of vertical bar plot. And so we, for example, infer about 40% of those mutations are neutral, about 60% are at least weakly to strongly deleterious. So basically 60% of the mutations in the very conserved regions seem to be deleterious. Okay. Well, we then did the same thing for using different bins and going down this constraint score threshold. And what you notice is, in fact, that we infer more neutral mutations, the light gray. That proportion goes up as we move down the constraint score distribution, which I think is exactly what you would expect, right? These are seem to be less constrained, some more neutral mutations, right? That makes sense. But you notice there's still a lot of dark gray here, in fact. and suggesting that there's some proportion, I mean, it's albeit smaller, but even if you go out to 30 or 35, even the middle of the of the distribution, you see there's still about 15% of the mutations are inferred to be pretty, at least weakly to moderately deleterious. And it's only when you go kind of out pretty far that this part starts to go away. And you might say, well, wait a minute, can you really detect 10%, being really deleterious? Is that actually going on, or are they actually under selection? And so we did, again, looking at some model selection here to see whether actually adding in a model with negative selection actually fits these better than a model without. And it turns out that once about maybe I'd say around here, the blue shows a gigantic improvement in fit. And it's really once you start to get to the green colors where the fit doesn't really improve. So I'd say maybe about 60 to 70% of the distribution, once you go down that far, you stop losing improvement in fit from adding selection. But that's still a lot of the genome that has a case where the adding selection to the model dramatically improves fit. And so what this in sum is essentially sums up to is about 18% of the mutations in this putatively functional part of the genome seem to be deleterious. And so you might ask, well, how well does conservation do at identifying the deleterious mutations? And so what I'm showing you here is essentially the CDF, or the cumulative distribution, of all of the deleterious mutations with increasing FASTCon scores. And so, for example, we would identify that about 20% of the deleterious mutations are captured in the 5% of the most conserved genome. And that maybe makes sense, right? There's an enrichment of deleterious mutations in these most conserved regions consistent with what you would expect. But you're looked at the other way, that most conserved 5% of the sequence is only capturing about 20% of the deleterious mutations. And in fact, you'd have to go out pretty far to maybe about 40 to 50% of the genome to be able to capture about 80% of the deleterious mutations. We've looked at this for other constraint score metrics in terms of whether the adding more species sort of helps with this. And the short answer is yes, it does. But the general pattern still holds, where basically the top 5 or 10% is only capturing a minority of the deleterious mutations. And so now this kind of brings up the third rail of population genetics, just this question of how much of the genome is under selection. That's kind of been a hot button topic for a couple decades now. And so we can play this game and do some back of the envelope calculations here. And so what I just told you was we infer about 18% of the mutations in the annotated functional states of the genome are at least weakly deleterious. But this annotated functional states overlap about 53% of the non-coding genome. And so doing some math here, simple math, we basically estimate about 9% of the non-coding human genome is under selection. And I think that's nearly twice as high as the commonly cited 5%, but is in line with other estimates and some newer estimates actually from deeper alignments from like Zoonomia, where they are coming up with that number, but not quite being able to use the constraint scores to actually identify which are the key sites. So in sum, that's what we come up with. So many mutations in non-coding parts of the genome are deleterious. Mutations in promoters seem to be more deleterious than those in enhancers. Mutations in phylogenetically constrained regions are more deleterious than those in less constrained regions. However, the most constrained regions are only capturing some of the deleterious mutations. And what this, I think, implies is that the genome is really a dynamic place with changing selective pressures and turnover, both from epistatic interactions as well as gene-environment interactions and other things that are basically not necessarily captured in a single score across deep evolutionary time. So in the last 5 minutes or so, I'm going to try to answer this question of whether deleterious mutations affect the fate of small populations. And I think there's sort of two complementary paradigms for what could happen with deleterious mutations, right? One is what I'm going to call the drift load paradigm, where you have small populations, these weakly deleterious alleles that I've told you are abundant, can drift up to high frequency, leading to a reduction in fitness. That reduces the population size more, which leads basically to this extinction vortex, and over long periods of time, you could have extinction. The other mechanism that I haven't really talked as much about today is essentially inbreeding depression. And the idea here is mating between close relatives creates runs of homozygosity, or tracts of identity by descent, and that exposes recessive deleterious mutations that can then lead to reduced fitness and extinction. And there's interest in which of these can be more important for the extinction. And the sort of approach we have advocated taking is shown in this conceptual picture of how to incorporate genomic information into conservation biology. Basically means taking genomic data, inferring parameters, kind of like what I was just telling you about, bringing in external life history data into computational simulations of the evolutionary process, and then basically look at the extinction risk, and then you can vary different scenarios and parameters to make some sort of management recommendation. And we applied this type of approach in studies of the vaquita porpoise a few years ago, where we wanted to see what the effects of inbreeding depression would likely be if the population under different management scenarios. And so these, really the key plots are up here, where we forecasted what would happen in the future if, in fact, mortality from getting trapped in fish nets was completely eliminated. And what you see is the population recovers really, really well. In fact, very few go extinct. But over here, if there's maybe only an 80% reduction in mortality from fishing, you see the population doesn't recover as much, and it has a higher probability of extinction. So this, I think is a great approach, and I really like it and advocate for it, but conservation biologists often don't really like that approach because it's very model heavy, model dependent. So could there be an easier way? And we kind of thought about this from staring at a number of species that we've worked on over the years in the lab in this sort of summary table here, where if you notice, the one that seems to have really strong evidence of inbreeding depression is the Isle Royale wolf. And there's extensive phenotypic evidence of skeletal abnormalities, failure to reproduce. The population is nearly extinct. And in prior work, we've looked at the genomic patterns of genetic variation in Isle Royale wolves down here compared to Minnesota, the mainland source population. And what you see is that you have these big stretches of high heterozygosity interspersed with regions devoid of homozygosity. And that gave us an idea about, well, what type of signal could we look for to try to see risk of inbreeding depression? And the basic idea, right, is we get long runs of homozygosity are created basically through mating among close relatives. And our working model for how they create inbreeding depression is that if you have heterozygous mutations in the genome, shown by these green stars, when you have a run of homozygosity from this inbreeding process, you essentially make the deleterious mutation homozygous and reduce fitness. And so that led us to a simple idea. Well, what if we try to take these different effects into account into a simple summary statistic that one could easily calculate from genomic data? So F-rho, or the amount of the genome in a run of homozygosity, that's something that can be calculated from genomic data, times the product of, or times pi, outside of the runs of homozygosity. And the idea is this is capturing how much segregating recessive deleterious mutations there could be in the population. And the idea is a population that's big for this would be more at risk of inbreeding depression. And so is this anything sensible to do? So we did a bit of a meta-analysis here where I'm showing F-rho here and heterozygosity outside of the runs of homozygosity, Different dots represent these different species that have been studied by us and others over the years, and the color indicates this ID risk statistic I was telling you about, with red indicating higher values, yellow indicating lower values and lower risk. And what I think is really interesting is that these species up here, like the Florida panther, the Isle Royale wolf that I just told you about, these are ones that actually do in fact have the stronger evidence of inbreeding depression, whereas Minnesota moose, Ethiopian wolf, vaquita, the ones down here, these are small populations that don't have extreme or don't have evidence of inbreeding depression, and they show up as lower risk with this metric. So in some long runs of homozygosity, which I didn't tell you the data for this, but numerous studies have been associated with lower fitness. We like modeling and forecasting, but that may not always be feasible. And so we propose this ID risk statistic as a simple yet informative summary to at least do some relative rankings of how concerned we should be. And lastly, putting kind of all of this together, deleterious mutations are abundant in genomes, and understanding them, I think, is vital for interpreting genetic variation. Studying variation in lots of species jointly can be powerful for learning about evolutionary parameters, but we have to be always thinking about new models for robust inference. And thinking about deleterious mutations in small populations, I think, is quite important and should be considered for management. So with that, I'd like to thank the current and former folks in my lab for working on this work, and once again, thank NIGMS for funding. I'd be, of course, happy to take questions. Yeah, George.
I have two questions related to your first part of the talk.
Yes.
So is there any hypothesis why body size would matter to S? And the second, so I wonder whether the mutational spectrum has been considered. Like, so if some species has more transversions than transitions, for example, and transversions presumably are more deleterious, they tend to cause non-synonymous changes, or tend to cause non-synonymous changes that will affect amino acid property more, then they could be more deleterious. So I don't know whether that factor has been considered.
Yeah, so great questions. The first on body size, I'm not sure, because usually for a lot of the molecular evolutionary type analyses, it's correlated with the effective population size, and then that's what ends up mattering. So I don't know, I don't really have a good idea on why that correlation looks stronger. So that's on that. And then on the mutational spectra, there is some evidence of it varying across different species. And so we did account for things like the CPG effects in the modeling of the mutation rate. But the idea that perhaps so not so much like what the rates are, but whether just there's certain types. So we did account for whether it's more likely to have a non-synonymous or a synonymous mutation in the different species.
But if mutational spectrum is different, for example, if a species has more transversions,
yeah,
and the transversions cause bigger amino acid changes, like
that could be. Yeah, that's right. And maybe that could actually be some of the more molecular ideas of what could be going on. Yeah, it's a great point. Yes?
Thanks for a fascinating talk. So along a similar line, I'm wondering if you can connect the beginning and last part of your talk for information. So thinking, of course, about the wolves, right? So they have a very large long-term effective population size, but the sample populations have a very small recent population size. So could that kind of dynamic with temporal changes in NES cause some sort of weird shift in your estimate of the average S over time?
Potentially. So for the wolves that we focused, well, one of them was the Arctic wolves that had a pretty big range and weren't like the sort of reintroduced or really like recently severely bottlenecked. So they didn't look really remarkable in terms of patterns of variation. That's not to say that there's that they've had a completely stable history over long periods of time. But that's, so I will say it seems that this feature is, canids look a bit odd, and we've looked at, like I showed you the two wolves, we've also looked at some dogs, and they look quite similar to the wolves. Which one could say is a surprise or not. I don't know which way to think about it, but it does seem that it's more canid rather than, say, wolf. Yes?
Yeah, thanks for the really interesting talk. I was wondering if you looked at, for the first part of the talk, genome size or other aspects of genome architecture, like, say, the bimodal chromosome sizes and things like birds.
That's a great idea. So one thing not yet, but that's on our list of things to actually look at in more detail beyond just the mutation rate. In mammals, there do tend to be more complex regulatory architectures and more protein-protein interactions than in insects. And so I think there is, we've tried, or I've thought of a complexity argument here, but the birds are kind of hard to figure out where they necessarily would fall in that. I think if you're comparing mammals to insects, I think you can maybe make those arguments more, but I don't know with how the birds necessarily fit in. Other questions? Yeah.
So I have a question. So when you're inferring DFE, you seem to assume that there seems to be an assumption that all the mutations, they are under constant selection, that those mutations are always, say, deleterious with S being constant.
Yep.
Maybe some mutations, they are sometimes beneficial, sometimes deleterious. They are under fluctuating selection or balancing selection. Will that have some influence on your results? And if so, is it possible to, using some similar methods to infer what's a fraction of mutations that are under fluctuating selection or balancing selection?
Yeah, so I think in terms of what that sort of fluctuating selection would do is, I think what we're capturing is some sort of average over that. Exactly how it's weighted is not entirely obvious to me, but I imagine we're capturing some sort of average effect. And then the other question about whether we could try to estimate it, in principle, I'd love to say yes, because that would be really cool. But I don't know if we would have enough power really to get beyond the average effect. I mean, maybe we would need some other features in the data to be able to try to do it. But that's an interesting thing to think about for sure. Well, thank you for the great questions.
That was great.
Oh, thank you.
It was very well delivered.
Thank you. Thank you. Thank you. And I like your question on whether the mutation certain, because it does vary.
I know it varies, but I don't know whether it has phylogenetic signal or not, because you have phylogenetic signal in your
Right.
Right.
Yeah,
and also, how would that be related to body size? I don't know.
Yeah.
So your body size results were very significant. 10 to minus 4.
That was the most
Yeah,
significant.
yeah.
So the other thing that we're going to do with NE is get a better estimate, because what I showed here is not ideal in that it's the same synonymous site one, and so it's a little confounded with
Right.
the getting the 2NS to the S, and so what we're going to do is take like non-coding estimates that are a bit
Yeah,
more
that's better.
independent to
Yeah.
use for the NE there, because I
Yeah.
think that's more
But I
appropriate.
suppose, so I was thinking about a connection between your first part and second part.
Yeah.
So if you were, I know you haven't,
Yeah.
estimated the non-coding selection in other species.
Ah,
Do you think,
yeah.
do you
Yeah,
think you will
yeah.
see a similar pattern, like the species where the S is bigger for coding region will also have bigger S for non-coding region?
I mean, my gut instinct is it might be reversed. Like, I might think that non-coding mutations in Drosophila might be more deleterious than in humans
Because
it's
more
more
compact?
compact.
Okay.
Yeah.
But I guess we
But if
will
we
have
took
to
the
control that.
Right.
Yeah,
so if we
yeah.
took the
Yeah.
functional parts,
Yeah.
right, that would be more of an apples to apples. Because I think the other thing with the enhancers being so weakly deleterious, I think those are harder to annotate.
Yeah, yeah, yeah.
And there's so much sequence
Yeah.
in
So
there that's
yeah.
part of perhaps diluting the signal
right,
a bit
right, right,
over
right.
the
Yeah.
not
If
as
the
tight
annotation,
annotations.
if the annotation is not accurate, yeah,
yeah,
then,
it's
yeah.
going to weaken
Yeah,
it.
yeah.
So
Yeah.
I think there's some of that for certain going on with the enhancers.
So there was
but
so you estimate 9%.
Yeah.
And I can't remember. There was a paper by
Yeah.
by Dan Grower
yes,
in
yes,
GBE.
yes, the televisions.
Yes, but he later he had another paper in GBE. So he was arguing about non-coding region,
Yeah.
functional non-coding region
Yeah.
cannot be more than certain percent.
Yeah.
The argument was if you have a lot of functional sites in the human
Yeah.
genome, given the human mutation rate,
Yeah.
then the mutation load is going to be too high.
Yeah,
So nobody can survive, basically.
Yeah.
I can't remember his estimate.
yeah. So I